Here we'll learn the key differences between meiosis and mitosis. Start a table to note the 6 key differences now, which we'll learn through this tutorial. Parent cell types, tetrad formation, crossing over, synaptonemal complex, kinetochore orientation, and genetic variability. Let's begin with the parent cells, the first key difference between the two processes. For mitosis, draw a somatic cell with 2 sets of 23 chromosomes. Indicate that it's diploid, 2N, 46 chromosomes. Write that somatic cells comprise most of the body's cells. For meiosis, draw a germline cell with 2 sets of 23 chromosomes. Indicate that it's also diploid, 2N, 46 chromosomes. Write that germline cells are reproductive cell precursors. Now let's illustrate the distribution of genetic material in each process. Indicate that DNA replicates in the S phase of mitosis and meiosis. This step is identical in both processes. Draw our somatic parent cell in prophase of mitosis. Show that each condensed chromosome pairs with its duplicate at the centromere. Now draw our germline cell in prophase 1 of meiosis. Show that the homologous chromosomes pair with each other as a tetrad, which marks the second key difference between mitosis and meiosis. We truncate the chromosomes for reasons we'll show in a moment. Paternal and maternal chromosomes are homologous when they contain the same genes in the same order, but contain varying allelic combinations. Now fill the truncated segment of the chromosomes with a segment of the opposite homolog. Label this as the chiasma, the plural is chiasmata, which is the site of genetic recombination. Indicate that this particular mode of genetic recombination is called crossing over, which marks the third key difference between mitosis and meiosis. This brings us to our fourth major difference, the synaptonemal complex. Let's take a closer look at the tetrad to illustrate this structure. Draw a portion of the tetrad, particularly the region in which paternal and maternal homologs join, as 2 rows of chromatin loops. Indicate that a zipper-like protein structure joins the homologs. Write that the synaptonemal complex, which is specific to meiosis 1, functions in synapsis. It holds homologs together and keeps them closely aligned until anaphase 1. Now let's draw metaphase of mitosis. Show that sister chromatids line up along the metaphase plate. Specifically indicate that the kinetochores in each sister chromatid pair face opposite poles. Thus, when microtubules depolymerize, they pull the chromatids within each pair to opposite sides of the dividing cell. Next, draw metaphase of meiosis 1 to illustrate the fifth key difference between these processes. Show that the tetrad aligns on the metaphase plate. Let's take a closer look at this tetrad. Draw the tetrad and include the chiasma. Now draw kinetochores on the outward-facing sides of the homologous pairs. In meiosis, the kinetochores of sister chromatids are adjacent to one another. Now draw spindle fibers that connect to the kinetochores. Write that the kinetochores of sister chromatids face the same spindle pole. This is different from metaphase of mitosis, in which sister chromatid kinetochores face opposite spindle poles. Next, illustrate that when the microtubules depolymerize in anaphase 1, they pull homologous chromosomes apart. Finally, show that after anaphase 1, telophase 1, and cytokinesis, the resulting 2 daughter cells are haploid. Indicate that anaphase, telophase, and cytokinesis and mitosis produce 2 diploid daughter cells. These daughter cells are genetically identical. Illustrate that meiosis 2 begins almost immediately after the first cytokinesis and produces 4 haploid daughter cells. Finally, indicate that each of these daughter cells is genetically distinct from each other and from their parent cell. This brings us to the final key difference between mitosis and meiosis. Genetically distinct daughter cells show that there are three facets to genetic variability in meiotic division: crossing over, which is genetic recombination, which we've already learned; random fertilization, a single male gamete must fuse with a single female gamete, which multiplies the genetic variability generated by independent assortment. And the third is independent assortment, right? That independent assortment refers to the fact that each tetrad positions itself on the metaphase plate independently of the other tetrads, which produces about 8 million possible combinations of chromosomes in haploid gametes. Let's visualize how now. Start with 2 cells in metaphase 1. In the first cell, draw representative paternal chromosomes on one side of the metaphase plate and maternal chromosomes on the other. In the second cell, draw representative paternal and maternal chromosomes on opposite sides of the metaphase plate. Now, below each of these possibilities, draw 2 cells to represent metaphase 2. Draw the alignment of chromosomes on the metaphase plate in each of these cells. Note that they are distinct from each other. Finally, draw 2 more cells below each of the cells in metaphase 2. These are the final haploid daughter cells. Fill in the chromosomes. Combined with crossing over during metaphase 1 and random fertilization, meiosis produces genetic variation within a given population. This marks the most significant distinction between mitotic and meiotic cell division. This concludes our diagram.